Svenja, and good afternoon. As we've seen in the previous talk and also in the wonderful symposium this morning, it's essential nowadays to try and get 3D cytoarchitectonic reference maps, especially in the human. And one way to do that, as was nicely presented just now, is by the alignment of many thin sections, and the big brain is a beautiful example of that. That shows you that at the same coordinate you can slice in different directions and look at the cytoarchitectonic layering in different dimensions. Now, this is a beautiful work that is very labor and time intensive, even in the specialized labs where they're capable of doing this. And even then, to get single cell level 3D alignment is possible, but extremely challenging. Now, there has been promise for a few years now that possibly optical tissue clearing, making tissue transparent, and then labeling it and imaging it with 2-photon or light sheet microscope could also help out there. And so indeed, in rodent brains, mice brain and rat brains, beautiful 3D phenotyping results have been generated. But in applying that to human brains, there's been a very big challenge still out there to apply that. So to summarize a few of the points there, in the much more myelinated human brain, clearing tissue and labeling it is extremely challenging, and so some studies have been performed in the pre-myelinated fetal brain. It's even more challenging in long-term formalin-fixed, or as we sometimes call it, archival tissue, which is, of course, a lot of the human tissue that is actually available to us. And so some studies have been performed in freshly frozen human tissue. And it is especially difficult in the target where we actually want to perform this kind of clearing and imaging, which is very thick, so larger than 1 or 2 millimeter tissue, and for dense labels such as cytoarchitectonic Nissl-like labels. So the rodent studies often use sparse labels, and many human antibody labeling studies have only managed to get up to about 1 millimeter depth, mostly because of these very large antibody molecules only getting so far into the tissue. So that makes all of this very difficult to scale to the pieces of tissue where we would actually like to use it. And that is also shown by the fact that the best available studies now, they can get up to kind of a cube of 5 by 5 millimeters, and even doing that takes them up to 1.5 years in order to actually clear and label the tissue. So we set ourselves the aim to try and see if we could produce a scalable clearing and cytoarchitectonic labeling protocol for human tissue. So the aims set out there are all the desiderata that we kind of set our challenges. We want large and thick archival tissue, up to 4 or 5 millimeter thick, because that's the cortical thickness, and that's where it really starts to matter that you don't have to do all of the alignment anymore. Centimeters by centimeters wide in the other 2 dimensions, because that's roughly the dimensions of where, at least in some parts, the lateral extent of cortical areas comes into play. See, for instance, here, this kind of semi-coronal cut through the calcarine sulcus that is running over here, and the Brodmann area 17 and 18, V1 and V2. It would require about 40 by 30 millimeters by 5 millimeter thickness to actually image that. But that would mean that, with respect to the current state-of-the-art of 5 by 5 by 5 millimeters, we want about 50 times larger. And since we want all of that time-efficient and cost-efficient, time-efficient being days or weeks, we essentially want to image 50 times larger than the current state-of-the-art in at least 10 to maybe even 100 times less time. So scalable is really the issue here. Now, with a lot of development, we've actually been able to come up with a procedure that can deliver on that, which we call MASH, multi-scale architectonic staining of human cortex, which is a solvent-based clearing approach and a small molecule labeling approach. And solvent-based clearing takes these steps here: pretreatment and permeabilization, labeling, delipidation, and then a refractive index matching with a refractive index matching solution. And with that, we can actually label very large and very thick samples, so 3 by 4 centimeters laterally and 5 millimeters thick, into about 10 days with dye cost only about $1 or €1 or even a Singapore's dollar per sample. And so you can contrast with that with the relatively high cost that you get for antibodies. So there's 2 essential innovations that we developed here that make that possible: the dyes in the labeling step and the refractive index matching solution, or the RIM solution, in the matching step. So for the dyes, we came up with 4 fluorescent small molecule labels, and that they're small molecules means that they're fast diffusing, and you can actually get them very deeply into the tissue. And we were able to develop 3 Nissl-type cell body labels in 3 different spectral bands, so that gives a lot of advantage in combining them. And especially the far red colors give you a lot of advantage in imaging very deeply with your light sheet microscope into the tissue. When it comes to the rims, you actually need to match your small molecule dyes with appropriate rims, because the rims that so far have been used in solvent-based approaches were very photo-bleaching and very corrosive. So they would bleach dyes like this within minutes, so that you couldn't even image them. And so the rims that we've been able to develop are very compatible with the MASH dyes. They're actually mixture rims, so they're a mixture of TDE and wintergreen oil, which were rims which were already used, but which in solvent-based protocols don't actually make the tissue very transparent, as I hope you can appreciate. But in adding cinnamaldehyde, CE, to the mixture and getting the proportion right, you can actually make 5 millimeter thick solvent-cleared human tissue very transparent and have several additive advantages, such as low corrosiveness, low melting point, and also even an adaptable refractive index through the mixture. So, if my slide wants to advance, yes, it does. On to some results. Here is our green cell body label, which is the acridine orange molecule, which binds RNA and DNA, so a very Nissl-like stains, which is able to give us very nice stains of pyramidal cell bodies, as you can see here in a 2-photon scan. And of course, it's able to do that into 3D stacks, as I'm trying to show you in these movies here. Here's some light sheet imaging in area V2, where you can see that we can go all the way in 3D through a stack of tissue and keep imaging all the way to the depth of the tissue. Showing some results then for the red label. If my slide wants to advance, yes, there we go. That's actually the neutral red molecule that is also a nice fluorescent Nissl-like marker, so label cell bodies. With that, actually, we can get quite deep. It has the wavelength advantage of having the red color, and we managed to actually get through about 5 millimeters of tissue. Here are some images imaged very deep, 2.5 and 3.5 millimeters. And although the Nissl red molecule gives a bit more of a sparse stain in the cell bodies, as you can see here, counter-stained with the DAPI for the nuclei, we can image very deep with it, something I can quantify over here. Typically in light sheet microscopy, there is an excitation direction of the light sheet coming in sideways into the tissue, so you'll get some signal drop off in that direction. But most of all, the problem is the signal drop that you get as you go deeper into the tissue, because the emission has to travel all the way through the tissue and there's a lot of scattering. But here you see quantified for a few depths. So here, 1.5 millimeter, 2.5, and even up to 4.5 millimeter in, that the cell body signal, shown in red here, stays very high, up to very deep into the tissue, whereas the background signal, which is in blue down here, stays very low, so that you have very nice delineation of the cell bodies very deep into the tissue. One of the best labels that we have when it comes to very deep imaging is actually the MASH-MB label, which is the methylene blue molecule, which has a far wide wavelength. And again, there we go, sorry, let me go back. Let's see if that movie wants to run while I continue. Which with that very far red wavelength, had the advantage of penetrating very deeply into the tissue. That movie didn't want to run, so I'll continue and show you another movie later. Then finally, we have matched to the MB molecule, the MG molecule, which is a very nice replacement for the UV DAPI or Hoechst stains for cell nuclei, and you can image them very deeply. And the ideal case is that you can combine it with the MASH-NR stain and get a double label for both cell bodies and cell nuclei. Now, of course, these labels, although they are already used in a bright field form for a Nissl-like stains, they deserve some validation, which we indeed did. Here you see a small thin section that we labeled with the NR label and the standard Crescel violet, and indeed you can see that the stains very nicely overlap and that you will get the Nissl stains that you're used to also when you image with the MASH labels. Then closing up, multi-scale imaging to see if we can actually manage to do very large and thick tissue, get cytoarchitecture of all layers, and do that in a way that is very time and cost efficient, and even try to get to deep cellular resolution 3D light sheet imaging. So for this, we took an entire human occipital lobe and excised 2 large pieces of tissue, 5 millimeter thick, that contain both V1 and V2, and try to image that at different scales as well as we can. So here you see some results from the more posterior thick-lined piece. Here it was before clearing, when it was just excised. Here it is after clearing. Indeed, you can see the rather large dimensions and the nice transparency after clearing. Here you see the double MG and an R label, V2 over here and V1 over here. And if we then zoom into V1 a little bit and actually turn that image upside down and actually label it in black and white and reverse the contrast so that you get a stain as an anatomist would be used to looking at a Nissl-like contrast, we can actually put that next to a reference atlas, such as the von Economo, and classify all of the layers. Going to the more posterior dashed section, the adjacent 5mm thick section right over there, again, we can zoom into this piece of V2 over here, and once again, we're capable of labeling all the different layers and see distinctive features such as the large pyramidal cells in layer 3B. And just to show you that that is actually in the context of an entire 3D stack, I will keep this running in the background here, and you can see that we can actually get cytoarchitecture at cellular resolution there up to very deep into the tissue. But zooming in a little bit into the field of view that I just marked in the previous slide, you can actually see, if my slide wants to advance, there we go, that we can go in a bit further. Here's layer 1, 2, 3, and 4. And in the context of that particular stack, which was a 3D stack, we can actually go in about 1.5mm in and go all the way in and find we could create a 3D reconstruction of a single pyramidal cell to actually kind of test the hypothesis that we can do cellular resolution imaging in 3D with this. So very briefly, my conclusion and outlook. We've got 3D cellular resolution cytoarchitecture on archival adult human brain tissue. It's very scalable in time and cost, and therefore very applicable into many labs. So this is really something that I hope is something that you could try in your lab as well and achieve data sets like this. In order to scale it even further, I have to say that imaging is already the bottleneck. So the data that I showed you in the previous slide was actually done at the limits of what current commercial light sheet technology can do. And in order to go even further, so in order to clear, label, and image these entire pieces of tissue, we'll need some advancement into the light sheet microscope. And also, as we scale even further, the cost efficiency of alternative labels such as antibodies will come even more into play. And of course, just as is the case for the big brain, as we produce these data sets, though, another thing that is definitely outstanding is the big data analysis of data like this. So with thanking my collaborators and the people in my lab that did this work, particularly Sven Hildebrandt and Anna Schutt that did the bulk of this work, I also want to thank you for your attention.